Thank you so much. All right, moving on, we have our GM1 board member, Vanessa Rangel Miller, here to talk about how to read your genetics report. Thank you, Vanessa.
Thank you. And I really appreciated the earlier talks, how exciting. Now we're going to talk about genetics, but I would really rather think about surfing and turfing and things like that. I'm Vanessa Rangel Miller. I am a volunteer board member for the Cure GM1 Foundation, also a genetic counselor. Let me see if I can advance the slides. I know we're running a little late, so I'll try and go quickly, and thankfully the earlier presenters also covered some of this, so I probably don't have to review quite as much. One more slide here. Okay. And so quickly, as you heard before, most every cell in our body contains the instruction manual for how our bodies grow and develop. Those packages of genes packaged into chromosomes. It's really that composition of DNA that gives the blueprints for the body. So we heard a little bit about that before. We also heard a little bit about how GM1 is an autosomal recessive disorder. Autosomal means the GM1 gene, which is GLB1, it's not affiliated or positioned on one of the sex chromosomes, the X or Y chromosome. So we have two copies of every GLB1 gene, or two copies in most every cell of our body. One copy inherited from mother, one copy inherited from father. And it's when both copies are not functional or not sufficiently functional that GM1 may develop. Now, the reason that happens is because there are changes, as we heard earlier, there are changes to the GLB1 gene. Those can be small, those can be larger. Those changes are often called mutations, especially in older literature. You'll see mutation used quite a bit. Alterations, change, variant. Variant is very common. That is the current terminology referring to it as variants. But we did talk about this a little bit, so I won't go too much more into detail. And what I'd like to do today is just give you a high-level example and discussion of what you might see on genetic test results. So first, we'll talk about the types of genetic changes that are often seen in GM1. You can see an example here, and as you heard about earlier, again, there's a specific code for the GM1 gene. That sequence is what genetic testing companies will read through. They compare a person's GM1 gene, the GLB1 gene sequence a person has, against what's expected. And they'll classify different variants along that process. Everybody has changes that make us different from one another, and many changes are tolerated and don't result in any health change. It's what makes us different from person to person. But sometimes changes really do impact the gene. You can see an example here, and this will hopefully help you think about your own test results or results from your family members. So if you think about a gene as being this example sentence, you can see the original one on the top. You can also see how that sentence can be changed and how we can apply a label to those different types of changes. So in the second sentence, we've changed the word hall to the word ball. That's an example of a missense change. We can see an example of an inserted word, the gray-green cat. That would be an example of an insertion change that might be reported on a genetic test result. Or a word missing. Say the word cat was missing. That's a deletion change that might be listed on a genetic test result. Duplicating the same word, cat, cat, again a duplication change, or entirely ending the sentence too early. That's an example of a premature stop, a nonsense change. So those are a few examples. There are others. Unfortunately, genetics is quite complex, so you can see different terminology used, but this is an example of what is most common. And so here I've made it an example test result. This one's not real. I put this together. Now, unfortunately, while you see the image of a test result on the left, they're not standardized. So there is no standard report format for genetic test results. Commonly, though, they do have the overall result at the top, or actually the person's information first at the top, and then just below that, the overall result. Is this a result that indicates disease? A list of the variants identified. There can be 1, 2, or more. Next steps from the testing laboratory, and then multiple pages often follow, which talk about the technical detail of the test method as well as the variant that was found. Oh, and I didn't realize I animated this, so there we go. Okay, so then if we dive into this a little bit more, you can see here an example. And you'll see some terminology, and we'll get into examples of this terminology. You'll see things like variant and zygosity, variant classification, and so that's where questions commonly come up. What does a variant mean? What is zygosity? What is a variant of uncertain significance? So we'll just talk through some of those examples, and then at the end or later today, I'm happy to take any questions if anyone has individual questions. So you can see here first, we'll talk about the gene. Again, we heard earlier, GLB1 is the name of the gene associated with GM1. If other genes are tested, they will also be listed on that same test result with their gene acronym listed. You can see here an example of a variant. And I've kind of tried to color code this. So typically, the variant's going to be listed in both the DNA or coding nomenclature, as well as the protein nomenclature. And you can see from the DNA nomenclature, it starts with a C, followed by numbers, that's the position of where the change is, then first the reference sequence, and then the change that is seen in the person. Whereas the protein nomenclature is a little different, starts with a P. You have the reference amino acid, then you have the position, and then you have the changed amino acid in this simple example. Zygosity, this is an example of this means whether the copies of the gene are the same or exist in different variant forms. So we heard before the term compound heterozygous. It means that the two changes are not the exact same. Homozygous means the two changes are the same, and that can happen. Both are quite possible in GM1. We also see classification, pathogenic, and I mentioned before, variant of uncertain significance. Pathogenic meaning likely disease-associated. And then if we get to the scale, so gene variants are classified according to current guidelines from the American College of Medical Genetics. It's a 5-point scale. You can see all of the categories listed here, but generally benign and likely benign you don't see on test result reports. You just see these 5. But variant of uncertain significance is unfortunately common. You saw Dr. Hastings present how often that happens before in the GM1 dataset. And this happens when the significance of a genetic change on the health or function of a person or a gene protein is not known. And there can be several reasons why this happens. Dr. Hastings pointed out that sometimes there's not enough information available about that variant and that impact on health. It could also be other reasons. It could also be that the symptoms a person has, or maybe the lack of symptoms, does not align with what would be expected for that particular disease. So there are a variety of reasons, but often it's data-driven that we don't have the ability to say, is this disease associated or not? So it's called a VUS. So lastly, inherited from, sometimes that's on results, sometimes it's in the fine print, and that just means whether parents were tested, was it found in the parents. De novo was a term we heard earlier. De novo meaning that change arose for the first time in a person, maybe isn't found to be carried by either parent. And lastly, one other thing that we've often heard on test result reports is whether the particular change or changes are occurring on the same gene, on the same chromosome, or whether they occur on 2 different ones. So if it's located on the same gene, on the same chromosome, that's called cis, in cis. If it's on 2 different ones, then that's called in trans. That can actually be helpful information. Testing the parents, testing family members can be helpful information for determining, is this a variant associated with disease or not? So with that, just a quick summary. I know we went fast, trying to save on some time here. But just again, the important things, one, genetic testing can be very expensive. Laboratories may offer testing programs that can help offset the cost, sometimes fully covering the cost based on eligibility. Sometimes that would include access to testing for parents or other family members as well. Definitely speak with your doctor or your genetic counselor about such opportunities because they do vary by lab, but also speak with them about your test results. So if you've had testing and you don't understand what has been listed, they're the great individuals to start to have that conversation. Speak with a genetic counselor. They can reach out to a lab. They can ask for more evidence. So I encourage that. If you are needing a genetic counselor, there's a website. The National Society of Genetic Counselors has a directory where you can find somebody near you. So with that, I will close, and hopefully I got us back on time, and I'll take questions as part of the break later.
Thank you.